Good afternoon, everyone. I think we can get started. Thank you for joining us. My name is Wenjin Ge. I'm from Regional HEOR. I'm moderating this session evaluating challenges in designing external control studies to contextualize effectiveness in single-arm clinical trials. So our discussions will be focusing on the hematologic malignancies. I am joined by a wonderful panel today that you'll be hearing from soon. We have Glenn Wu from Janssen Scientific Affairs, my dear colleague, Kristen Han from Regeneron, Paul Spin from Avazana, Arnold Chen from TrinetX, and Catherine Leroux from the Oncology Center of Excellence of the US FDA. Some background on the topic, the ECT and ECAs, I think many of you are very familiar with. So in an externally controlled trial, you compare patients receiving the test treatment to a group of patients who are external to the trial and who had not received the same test treatment. So we refer to the second group as the external control arm of the ECA. Single-arm trials are commonly implemented in rare conditions, and which is oftentimes the case for oncology, especially hematologic malignancies, because they are often more rare than solid tumors. And we have been seeing increasing use of the ECAs to contextualize the single-arm trials in oncology, and they can be used for different purposes. In addition to regulatory submission, it can be used for HTA submission. It can be used for informing internal decision-making and clinical practice and treatment guidelines. I listed a few facts here. So among all the 11 oncology approvals by the FDA in 2019, 6 of them included a real-world evidence study, but only 2 were considered supportive. On the EMA side, among all the cancer drugs approved between 2016 to 2021, 17% of them used some format of external control, and half of them were in the hematology side. But 37% of them were not considered even supportive by the EMA. And lastly, among all these single-arm trial-based HTA submissions in the last decade, half of them used some format of external control data. So as we are seeing more animal ECA studies being done, but I think we are still together exploring the best practices of doing them. Maybe you have seen the guidance that's released by the FDA earlier this year on the conduct and design of ECAs using individual patient-level data. So the guidance provides some clarity on the scientific rigor expectation of these ECAs. It also reminds us the challenges we're facing of doing the ECAs. For example, you need to pre-specify the ECA ahead of the clinical trial conduct. So it's basically has to be integrated into the trial program. And there is also the differences between the real-world data and the trial that you have to take into consideration in terms of the data availability, the patient characteristic variations, the assessment of the outcome in the real world may be completely different from how you do it in the trial. And last but not least, there's uncertainty in the regulatory acceptance and the validity of these ECA findings. So in this session today, we wouldn't be able to go through all the challenges. Instead, we picked a few very specific topics in the hematologic malignancy area, which is very interesting to me personally. Some of the challenges are very unique or more prominent for this area. So you're going to be hearing more from the panel soon. We have 5 presentations in the next hour. So depending on the timing, we may be able to take a 1 question by the end of the presentation. If not, please stay with us till the end. We have time saved for the discussion, the QA. Please stay with us. So we really look forward to hearing and learning from all of you. Without further ado, I will pass to our first speaker, Glenn Wu. He will talk about the ECA data source considerations in multiple myeloma.
Thank you very much, Wenjin, and thank you very much, everyone, for coming. How are you doing today? Yeah. So I'm feel very privileged to be invited in this panel talking about this very important topic. because ECA has been very commonly used in the hematologic malignancy space. So today I'm going to talk about this topic just from the data, selecting the appropriate real-world data perspective and in the context of relapse refractory multiple myeloma, because this is the area that I'm been working in during the past couple of years. So everybody knows that the relapse refractory multiple myeloma is a rare disease, especially in those later line settings. After patients progressed on the disease for a number of lines, the patients are really, under a very poor prognosis. So, the disease has a profound unmet medical needs. And also in these later line settings, without those innovative therapies, those patients have no treatment options at that time. So what they all do is, physicians are trying their magic, trying to recycle those existing therapies over and over again. So the outcome of those patients are really poor. So just to name a few, data that on average, the ORR in those later line settings is about 30%-ish, traditionally, and also the PFS is about 3 to 4 months. So really it highlights the unmet needs there, not only from the poor prognosis, but also lack of option at that time. So I think our regulatory, agents, not only FDA, but also globally HTAs, they all recognize this need over there. So they grant accelerated approval to those big new wave of innovative therapies. I just listed several treatments that were approved during the past few years to CAR T therapies and the one bispecifics. And just this month, there are 2 more bispecifics launched in this space based on single trial, a phase 2 single trial. And one more thing I wanted to highlight here is that because this new wave of innovative therapy really brings transformational efficacy, that it almost more than doubled the efficacy in terms of ORR to those patients. So it's really not ethical sometimes to run a head-to-head clinical trial to compare those innovative therapies versus the standard of care. So all of those, brings to the result that ECA becomes a very pivotal tool in this space to demonstrate the relative effectiveness and safety versus the competitors. So before we dive into the choice of real-world data, I think first let's step back and take a quick look at what a typical phase 2 single-arm trial looks like in the relapse refractory multiple myeloma setting. So from the eligibility criteria perspective, normally we will require patient to have the disease, progressed on certain number of lines, right? And also require patients to have drug exposure to a certain number of classes like the PI, AMID, and anti-CD38 antibodies. And also requires the patient to have some evidence of progressive disease, as well as the ECOG score about patient performance. And also, very commonly, you will see patients with some renal dysfunctions measured by certain, lab results like creatinine. Patients normally are not eligible for this trial. From the endpoint, perspective, because it's a phase 2 study, and the goal is to have the drug being approved in a relatively, timely manner. So at the time of the initial data submitted to the agency, the normal follow-up is about 9 months. So really, the primary endpoint is not going to be PFS or OS, that as we traditionally see in the solid tumor space. So ORR is very commonly used in this space as the primary outcome. So among secondary outcome with longer follow-up, we will also report the PFS, OS, and also depth of response measured by MRD negativity, as well as the incidence rate, severity of AEs, and also patient experience measured by patient health-related quality of life. So these just to show you all the important clinical data that the clinical trial collects. Okay. Now let's look at what is an appropriate comparator ECA in this space. If you recall at the beginning, I said there's a huge unmet needs, right? There's no standard of care. So very commonly that the external control arm that we use is a cohort called the physician's choice. Basically, it represents a mix of treatment regimens at the given time that the physicians may choose to the population due to different factors. Now, there are two things we really need to think about the data physician's choice cohort. One is the treatment paradigm has been, evolving very rapidly. If you recall, Dazarex was launched back in 2015, right? And since then, it moved from the later line setting to the frontline setting. So it totally transformed all the line of sets during this time period. So, you needed to make sure that the ECA that you captured is really matched with the clinical trial. So in this hypothetical example, you see if we have 150 patients enrolled in the trial from 2019 to 2022, you wanted to make sure that your ECA covered that period. And secondly, I wanted to highlight is that these, quickly evolving treatment landscape is not just a dynamic, but it's also diverse across country, right? As there are many factors, including, treatment guidelines differences, but also, HTA approvals. Certain regimens may not get approval in certain countries. So it's very important that you have this global footprint in mind, okay? Now let's look at what's the available data, in the industry available to us, right? So for pharmaceutical companies who had a big portfolio like Janssen, we may have a trial for Dazarex, right? We may use those trial data, using the long-term data, use them for to create an external control arm. However, for today's purposes, we were just focusing on the real world data. And people in this room are all experts in this space. So what I want to say is just a very general, perspective. We know there are typically 4 types of real world data that we can have in the industry. One is from the payer claims, like Optum claims, right? Second are the electronic medical records, and medical record is just a general term, but how the data was collected, there's actually, it's very heterogeneous in this space, which we can dive a little bit deeper later. Then, chart review study is another way to collect the data retrospectively by reading patients' charts, which in this approach, you can capture a lot of the, valuable clinical data that those secondary structured data won't be able to give you. One example is the Mammoth study, which has been commonly used for, external control arm or ITCs. And the last but not least is probably the most rigorous data collection method, but also is the most expensive, data type or registry data. One good example is the local motion study. So far it has been, several ITCs using local motion for the external control arm. So this slide just shows you a high-level framework of considerations that we need to think about when we select appropriate data. I want to get into all the details, but I just wanted to highlight a couple of thoughts, right? When you think about the limitation, really don't think about it as a dichotomous yes and no. For example, do you have ICD code to identify comorbidity? Right? It's not a dichotomous, question. Really think through how the data was collected, right? Whether or not the data that you collected has unbiased estimates or, estimates of the prevalence of the disease. Give you an example that I encountered recently. Imagine if you are a patient meeting with a specialty hematology in this space in their office. In your conversation with your physician, the physician is more likely to ask you about the disease-specific conditions, whether you have anemia, right? Whether you have, renal disease, or whether you have hypogammaglobulinemia, right? But they will not discuss if you have depression or, diabetes or hypertension. So those general chronic conditions probably are less likely to be documented in those EMR data. So it's not whether or not I have the ICD code to capture it, but also, think about whether it is complete or not. One more thing I wanted to highlight here is about the generalizability, which is very important. I wanted to highlight is that it's not just about the sample size or geographic representation. It's also about whether the data can appropriately represent the practice setting, that to make the data comparable with your clinical trial. Because trials are often done in the medical centers, but in the US, a lot of patients are treated in the community settings, and the way community treat their patients is very different than the clinical, than the academic settings. So you want to also keep that in mind. Okay, and my colleague later will talk about the challenges in the outcome, clinical outcome, how the methodology to address that. So I want to dive deep here. Okay. And I know everybody like Amazon rating system, right? So what I do is keep it simple. Don't do too much data, right? Let's just do qualitative, high-level assessment based on my experience during the past decade. So following the same framework, let's look at the relative strengths and the limitations across these 4 data types. So in general, no-brainer, registry gives us the best data quality, especially with the data elements needed to make a comparable cohort, with the inclusion/exclusion criteria variables as well as clinical outcome variables. However, the cost is so expensive, right? We have to think about other alternatives. From that regards, we think chart review probably is the second best choice, and structured, payer claims and EMR probably is the cheapest option, but it has the biggest data challenges. And these green boxes I highlighted there is speaking to the relevant strengths relatively between those data types. So with all these, things in mind, right? Let's think about what should we do with these data, right, in the industry? So based on my previous in the relapse refractory multiple myeloma space, what we did is we use number of real-world data to complement each other. So basically, you are really create a basket of data, right? Generate the ECA multiple times, so the underlying population from each data can complement each other. And one data set, in this hypothetical example, right, we can have a data from a large community practice, EMR, from the US region, right, but mainly collected from community settings. But from the endpoint perspective, you may not have the ability to check the ORR, but you have PFS, OS data. To complement that, right, we can work with a global, patient registry like a Locomotion, which has a global footprint, have a heavy academic representation, and also provide endpoints on ORR so that they can complement each other. One last step before my summary is that oftentimes when we decided which data to use, it's a very good practice for you to list out all the important variables and then rank them, relative to their importance to your study, right? And then check their availability and quality across the database that you use. In this hypothetical example, you can see, we listed about 15 variables over there, and then check the data quality and, availability across the 3 data sets. Okay. So in summary, so based on my experience from industry perspective, I think, at this point, administrative payer claims probably is the least favorable data sources for ECA. It's just because in this space, it misses so many, clinical data that it's really difficult to conduct an ECA, based on surrogate endpoints. EMR is very heterogeneous. Some EMR may be collected from, a specialty practice, which really is preferred because it may give you the data that you need. And also, if a data vendor can provide you with the data through its data curation process, that they can clean the data for you, that is very, very helpful. And one more thing is, there are some vendors out there, when they provide EMR data, they may have the ability to do ad hoc chart review, which enable you to create a small cohort of patients to run validation studies. This will hugely improve your study credibility. Okay, so that is definitely preferred. And then, regarding chart review and the registry, I combine them together because from the data quality perspective, they can all provide you what you need. Of course, those data sources covers multi-country, multi-centers, and multi-practice settings are preferred. But the cost is a big issue, especially to pharmaceutical companies that may only have one asset in this space. This investment may not be an feasible option. So here, I think this is a great opportunity for me, representing industry, make a call, right? Really call for unite, a collaboration across our regulatory team, industry, as well as guideline groups to co-sponsor a natural history patient cohort so that we can not only create that, but also maintain it so that all the players can reference to the same benchmark in the long term. Okay, thank you very much.
Good afternoon. I'm pleased to be here, and I'm glad that so many of you are in attendance. My name is Christian Hemp. I work for Regeneron Pharmaceuticals. I will talk about treatment response in clinical trials, in the real world, and what issues arise when they don't match up. Here's how a clinical trial may ascertain response to treatment. The example that I chose is for diffuse large B-cell lymphoma, a hematologic oncology, where treatment response may be ascertained using scans. You see some scans on the right side. You see lesions on the panel A, fewer lesions or none on the panel B. So this is a patient who responded to treatment. You would typically have a scan at baseline before the patient initiates treatment. And then you have a schedule of scans, for instance, at month 3, month 6, that are documented or prescribed in the protocol and then ascertained for all patients. This is the idea. Even in clinical trials, we're not going to have all the scans, but we should be pretty close to this ideal. The example that I'm using is for DLBCL, where, as I mentioned, response is ascertained through scans. But the example really extends to all kinds of cancers, solid tumors included, including those where the response may be ascertained using labs. It's essentially a missing data problem that I'm going to talk about, so it's not just limited to scans. So then how we go from scans to actually calculating response. So for every patient, we have a baseline and a follow-up scan or multiple follow-up scans that will then go through an independent review committee that reviews the scans, and based on those, determines response. Response may be categorized into these categories. This is simplified. Some sponsors use additional categories. So you can either have complete response, that means a complete disappearance of lesions, partial response, that's a shrinkage of certain lesions by a certain size. And then you have various flavors of non-response. So the patient may have stable disease, progressive disease, and then various flavors of missing. And that's what I want to focus on today. So missing or non-evaluable response may include scans not done, scans done but not available, scans not readable, or the patient dying before the patient could have been scanned. Now, based on those in a clinical trial, you would calculate objective response rate, ORR. In the numerator, you have patients who responded. That's CR and PR in this case. And in the denominator, you have all patients who started treatment or were randomized. And in a clinical trial, per protocol, this includes patients who have not evaluable response, missing scans, non-readable scans. In a clinical trial, especially in a single-arm clinical trial, these count against you, because these are considered patients who did not respond per this calculation, even though they may have responded, but we don't know about it. Now, how about real-world data? Here are 3 example patients. The first patient may have a baseline scan and a follow-up scan, and there may be a recorded physician-assessed response. That's assessed by the treating physician, not independent. The second patient may not have a baseline scan, may not have a follow-up scan, but only a physician ascertained response. The physician may have had access to scans, you may not, or they determined response on some other factor. The third patient doesn't have any response assessment and passes away before he or she would have had a scan. Now, what do we do with them? There are multiple options to ascertain treatment response in real-world data in an external control arm study. If you want to closely mimic what was done in the clinical trial, you could also do independent central review of scans. This would mimic the trial, most comparable, but it could only be done in patients with scans. And if you want to power your study accordingly, so if that's your primary analysis, you need more patients. There's a potential for selection bias. I will get to that. And of course, there are cost and timeline implications of obtaining scans and of reading the scans. If you go that route, what you can do in addition, you can look at treating physician assessed response in a superset. So you could ascertain scan response in a set of patients with scans, and then in a larger set, physician assessed response. Alternatively, you could primarily rely on treating physician assessed response. So you forego the independent central review of scans. These typically have wider availability. It's not independent, it's recorded by the treating physician. And it does deviate from the target trial methods because the target trial does independent central review. In this particular case, you could still do independent central review in a subset of patients who have both scans and physician assessed response, and you could try to validate physician assessed response. The risk that you're running is that it doesn't line up. So you may find late in your study that there's inadequate concordance. Another approach is you do both where available. So if patients have scans, you do independent central review of scans. If they don't have scans but physician response, then you use physician response. The downside here is it's not uniform, so you have a different assessment throughout your cohort. And there are similar considerations to what I mentioned under number 2. If you do a validation and the concordance is inadequate, then number 3 is not ideal either. This is not comprehensive. There could be additional ways to do this. One is, for instance, to forego ORR entirely and just focus on OS. You have a hard survival outcome. That's something that avoids these issues. But as Glenn mentioned, the primary outcome in your study might be ORR. You may not have long enough follow-up for OS ascertainment. So you kind of need to address ORR. For the remainder of my talk, I will focus on the first option. So let's say we did decide to do independent central review on scans for all patients. Now, what are the implications of different availability? In this hypothetical example, I'll use the mouse. Yeah, the mouse doesn't work either. Okay. Maybe. Oh yeah, there we go. Good, good, good. Okay, this is hypothetical. And what I wanted to show here is a trial, the target trial, and your real-world cohort. And the way I set this up is that the ORR is the same. So this is under the null, which in a real-world example should not be the case. You wouldn't do the study if your drug doesn't have a chance of beating the external control arm. But in order to illustrate bias, having equal percentages here does the trick. So this is designed such that both have a 30% overall response rate among patients with response available. And when I say response available, it means response information available. They could be non-responders, but you need to know about that. If I calculate ORR according to the clinical trial protocol, in a situation where in the clinical trial there are 5% not evaluable, and in the real-world data set there are 30% not evaluable, then I will have a bias analysis. So this is my ORR according to the clinical trial formula that considers not evaluable in the denominator, but not in the numerator. So I underestimate response in my external control arm study. In fact, I do underestimate response in both, but it's differential. And what does it do? It biases my analysis in favor of the clinical trial. So I underestimate response primarily in the external control arm study. So it's not the conservative thing to do. The alternative is to deviate from the clinical trial protocol and exclude the non-evaluables either from both or from the real-world data set. So in this particular case, I exclude these and I calculate ORR out of the responders and then the non-responders and those who died without scans. So this would go fairly close. And you may or may not do that in the trial, depending on whether you are able to do it. But the trial bias should not be as pronounced because you have much more completeness. Now, am I really telling you to condition on available response, conditioning on the future? Well, at ICP last year, we saw a poster that was very well done that showed that if you condition on available response, you introduce immortal time bias. And that should be to no surprise here, because if you require that follow-up scans available, patients have to survive until they have a scan. So you exclude patients who die without having a scan. So what we recommend doing is, if you do condition on scan availability, you need to allow patients to die. So we would include patients who died without having a scan. We are still conditioning on the future, even though this mortality issue may be addressed this way. So we do want to find out the potential extent of our operations. So it's important to do a sensitivity analysis that does not condition on response, on available response, I should say. But we saw on the previous slide that that's a biased analysis, but at least it gives us some sort of boundary here. We should also do a secondary analysis that looks at treating physician-assessed response. As I mentioned earlier, this could be a superset of patients, so we don't have that selection factor if we do that. And we could look at treating physician-assessed response in patients who also have scans, and treating physician-assessed response in patients who don't have scans. So that could tell us if the scans are available selectively or if it's pretty much missing at random. Okay, good. All right, ORR, but how about overall survival? Obviously, calculation of overall survival does not require follow-up scans. If you do condition on available response, so we have the same cohort as we have in the ORR calculation, and now we calculate OS in that cohort, we would selectively exclude patients without available response, but who survived. Because we are dropping those who died, right? We drop patients who died and don't have scans, but we now include patients, or not, we exclude patients who don't have scans and who didn't die. So we remove them from the denominator. That means the remaining portion of people who did die now becomes a larger proportion. So we overestimate mortality. So the recommendation here is to calculate OS both with and without conditioning on available response. Something that I want to mention is very important here. When you do condition on post-baseline information, this is done at the time when you are still building your cohorts, because the availability of a scan now becomes an inclusion criterion. So we are using post-baseline information at the time when we build our cohorts. It's very important to maintain blinding here. It's very easy to accidentally come across the proportion of people who died, which is one of your study outcomes if you look at OS. So it's important to keep those patients aggregated, at least when the data are presented to you, so you can't infer OS. You may need to establish a firewall among the different actors. So between CRO, scan vendor, and sponsor, there should be a firewall in place so that the sponsor only knows about OS or can infer about OS at the very end of the study when we really produce results, not at the time when we build our cohorts. To summarize, ICR of response information in real-world data can maximize comparability to target clinical trial methods, but it may necessitate conditioning of patient inclusion on available baseline and follow-up data. When you do that, patients should be included if they died without having follow-up scans. Conditioning patient inclusions on available response data deviates from trial methods, but it is the conservative thing to do, because otherwise you're biasing against the real-world data and toward your drug. Calculation of OS should be done without conditioning on response availability. And then again, maintain blinding. Thank you.
So I'm going to talk today about approaches to selecting time zero in external control arms. So this is a bit of a nuanced and technical topic. So I've presented some stylized examples to help walk through how we typically go about doing this. And I think it follows nicely from some of the general discussions on how you select data sources and approaches around handling outcomes. So my name is Paul Spin, and I'm an employee at Eversana, which is a life sciences consulting firm. So why do we really care about the problem of selecting an index date? Well, first of all, we need to define what we mean by an index date. So generally, an index date or time zero is the start of follow-up relative to when a patient meets all eligible inclusion and exclusion criteria for a trial. So in a single-arm trial, you might have a screening period where the study determines whether or not patients are actually eligible for receiving an investigational agent. And then once they're assigned to the treatment or once they receive their first dose, that's when you start to count data towards the outcome. So that's when you start to measure OS, PFS, when you start to look for scans over the follow-up period in order to evaluate response. So typically in a clinical trial, there's one line of therapy, there's one investigational agent, so it's fairly straightforward. You have one index date, one time zero. Typically, in an external control arm trial, using, let's say, retrospective EMR data, or perhaps you have access to registry data, or any real, even administrative data, although that wouldn't really work well in oncology, you tend to have rich longitudinal information on the patient's treatment journey. And so that means you can really string together lines of therapy. So in reality, when you look at this real world data, you could have 3 lines of therapy or 5 lines of therapy for a patient. And so there becomes a question of, well, which line of therapy should we choose to start measuring data over follow-up? So that's really the essence of this talk. And I've put together a hypothetical situation that kind of reflects the stylized example that I've experienced over the past 3 or 4 years working through these types of studies. It applies to the indication that we're talking about today, but generally it applies across many different indications, both in oncology and non-oncology. So here we have we're assuming that we've gone through all of the examples of different data sets. We've picked one that's fit for purpose. We know it's fairly high quality. And we have 2 variants of patients. So one has 3 lines of therapy and another has 2. And across all of these lines of therapy, the patient meets all of the inclusion-exclusion criteria that are applicable to the trial. And so we're fairly confident that these patients at these lines of therapy are adequate proxies for what we would expect to observe in the real world had these patients not received the investigational agent. Okay, and along the top, we have 4 standard approaches for picking a line of therapy. Okay, so we'll go through each of those and kind of highlight some of the advantages and disadvantages. And then I'll summarize and we can probably discuss a little bit more because I'm sure there are many in the room who have experience with this as well. Okay, so the first idea is probably the simplest. If you have a collection of lines of therapy where the patient is eligible for the target trial, you would just pick the first, right? Very easy. So you pick the first line of therapy. Once the patient starts the standard of care at that line of therapy, that's when you start measuring endpoints. So this is really attractive because it's simple, it's intuitive, It's very easy to implement. So Christian had talked about ICRC adjudicated response. As some of you who have maybe designed chart reviews in the past may very well know, when your chart reviews are quite laborious. So if you're extracting information from charts and you need to go and also adjudicate that with a group of physicians, if you have 700 lines across a cohort representing all available lines versus just 100 or 200 all representing the first line of therapy, you can get adjudicated responses much faster just using one line of therapy. Okay, and the other, I guess, technical point is when you pick the first line of therapy, you're not sampling those lines of therapy conditional on anything that happens after the patient enters the cohort for the first time. So there's no risk of selection bias. But the disadvantages of this are really two things. One is you're picking patients earlier on in their treatment journey, and that may not adequately reflect the types of patients that are being recruited into a trial, which may come later after patients have exhausted certain standards of care. Okay, and then the other bit of information is that you're discarding potentially very valuable data across other lines of therapy, and that can limit your precision and your power. So if you're dealing with a rare disease where you don't have very many patients, or maybe they don't live very long, so they don't get very many lines of therapy, this can be very problematic. So that's definitely something you want to consider when you're choosing between these four options. The second approach is really the converse of the first, where you look at the patient's treatment journey, you identify the last line of therapy, and you pick that line of therapy. And that's where you start measuring all of your data. So again, very easy to implement once you have a data set. You can go through abstraction fairly quickly. You can go through adjudication of the response with a panel of clinicians very quickly. Some of the same limitations arise as with the first line of therapy. So you may lack overlap in the line setting relative to your clinical trial. You're discarding other eligible lots, limiting your precision and power. But most importantly, I think in this case, you need to observe the patient's full treatment history. You need to, or I guess, treatment journey. So you need to know how many lines of therapy they received, and that can introduce some selection bias. And in practice, what you're doing is potentially picking patients artificially closest to their date of death, which can be associated with reduced response. So definitely, there have been many academic studies simulating different types of sampling procedures, and they're quite unanimous in arguing that this approach should generally be avoided. Okay? And then another equally straightforward approach is just to take all available lines of therapy. Okay, so here we take for the first patient who has 3 eligible lots, we utilize all of their information. For the second patient with 2 eligible lots, we again utilize all of their information. If we're looking at OS, we would be measuring OS from the start of each line of therapy. So really for the first patient, we would have 3 observations. For the second, we would have 2. And so 5 observations in our data set overall. Okay, so this is nice because it leverages all available data, which increases the precision of your estimates. It also gives you more power to detect an effective interest relative to your trial. Because you're selecting all lines of therapy with equal probability, in this case of probability 1, which is independent of available lines of therapy, this avoids the type of selection bias that I was describing earlier. Okay, thanks. The disadvantage of this is you require more time for data abstraction with your clinicians. So again, going back to that example, if you have 800 lines, that can take up a lot of time with clinicians, and clinicians have very high stipends, so it can be both time consuming and expensive. And multiple observations can be correlated within patients, so you require slightly more complex technical methods in order to get your parameter estimates correct. So I'll kind of skirt over that for today's purposes. And then for the final approach, the random line of therapy, here it's kind of basically a sampling procedure where you select one line of therapy with equal probability across all available lines of therapy. So in this example, for the first patient, we selected the first line of therapy, and for the second, we selected the second. And then those lines of therapy are used in our analysis. So again, we have faster data collection abstraction. We can actually make the sampling process very flexible. So, it's possible to weight our patients so that when we sample, the sampled patient population matches the target trial in some way based off of baseline characteristics. The issue here is that we were discarding certain eligible lots, which limits precision and power. The choice of therapy does partially depend on available lines of therapy because you need to know what range of lines of therapy are available so that you can sample from it. And in this very simple example, the estimation does not really account for the uncertainty in the sampling process. So you would really, in order to adequately capture that, you would need to bootstrap the process over and over again so that you can potentially sample different lines, not just the first and second as illustrated on the slide. Okay, so just to summarize again, so ECAs usually capture multiple eligible lines of therapy, which can lead to multiple choices for time zero. So when feasible, leveraging all lines of therapy is generally preferred. It gives you the largest sample, greater precision, greater power. It avoids lack of overlap in the sampled line setting versus the trial. But as I mentioned, it may be necessary to select a single line of therapy if you're trying to augment the data set through some form of expert clinical review and adjudication of your endpoints. And so in that setting, usually selecting the first line or a random line would definitely be preferred over the last line. So I think I'm at time. I'll pause there and hand things over to my next colleague. Thank you.
So first of all, I'd like to thank the organizer, Wenzen, who put together this symposium. And it's delighted to see folks sitting in the back, also standing in the back. So we apologize that we don't have a larger room. Okay, this is my disclosure. So first of all, I would like to say that as the chair mentioned, that the intention is for us to pick one topic, not to give a very wide overview. And in the two previous speakers, they pick a topic and then actually they provide a solution. Okay, the different line of therapy, different options and how you deal with it, measuring outcome, and then which may be the best options. For me, I'm not going to give a solution. I'm just going to highlight a problem that we face. So apologize, I'll chicken out. And then I won't use up the whole 12 minutes because I do think we all want to hear what the FDA would like to tell us about the regulation. So let us then get started by putting it into a target trial emulation. So what I'm saying is that actually it's not that different. The heterogeneity of the comparison group is actually not that different from a clinical trial. All right, so we face the same issue. And it is actually an issue that the first speaker mentioned already. It's the heterogeneity of your comparison group. Okay, so ideally we have someone on a standard therapy. And then you add, because hematologic treatment is, hematologic malignancy is very complex. So you just it's unlike that you have a sprained ankle and then you take some painkiller. So you have a lot of standard therapy already, and then you add a drug A, and then you have a new drug, and therefore it will be drug B. And so it could be a placebo in an active comparison. All right. So, but the challenge I'm going to use maybe the next 3 minutes, and I imagine all of you have some experience already, is that, what is standard of care? So we use the term SOC all the time, but is it really a standard, consistent SOC across time and across region? So that's that's actually the end of this topic. Is there a lot of heterogeneity? So I'm just going to highlight, what are the real issues involved in those heterogeneity? Okay. All right. basically it's a moving target, right? Okay, so for example, I went to school, went to medical school some years ago. Learned that, okay, there are two types of lymphoma. So there's non-Hodgkin lymphoma, there's Hodgkin lymphoma, right? So me being Asian, I learned that, okay, non-Hodgkin lymphoma is actually more common among, patients of my region. And then among the Western world, Hodgkin lymphoma is more prevalent, all right? So that was 30 some years ago. But of course, we fast forward now, then there's, you have B cell, you have lots of, biomarker that help you differentiate, the lymphoma into different subgroups. And, that is only one, there's only one disease, that's a lymphoma. And then if you apply that to leukemia, you have to apply it to lymphoblastic leukemia, the myeloblastic leukemia also, and so forth. So then all of a sudden, then, a disease, then every single subtype become an orphan drug, because, they're so rare in this country that it will actually qualify, as an orphan drug. So that's one dimension of the complexity and the heterogeneity. And because, there's an orphan drug, there's a business, there's a market for it. And therefore, drug companies are developing effective therapy, for these different subtypes. So then all of a sudden you have multiple companies developing, multiple treatment, for all of these subtypes. Okay, so that's another dimension. And then the last complexity is actually the line of therapy kind of highlighted by my previous speaker, is that, you have a new drug, when you develop it, it was for the last line. But then you gain more data, you design better trials, and then you move up to the second line, and then you move up to the first line. So if the SOC is the original last line therapy, and then all of a sudden that drug is not being used for that, only exclusively used for that condition. So the line of treatment varies over time. And then, of course, there's a solution, proposed by my previous speaker. All right. And then lastly, that's my experience. So last 10 years, I work in East Asia. So I observed it firsthand. And both from a trial design perspective and also from a reimbursement perspective, because both of them actually have implication for defining the standard of care. So a drug may be developed in the Western world and then being utilized, and try to seek reimbursement in some countries in East Asia, Southeast Asia, Europe, many parts of the world. But then you may recognize that the insurance coverage may differ. So I sat in a national committee to decide on insurance coverage, in the place where I previously worked. So we commonly see that, okay, this drug is developed, by a big company in Europe, in Western Europe and North America. So this is the indication. But, in a certain country, with limited insurance budget, there may be additional restriction put on top of that. So therefore, in that country, the SOC is very different from the SOC in the United States or Western Europe or Canada, or also Canada, right? So that's something that, I don't have a solution, but I just want to point out that, that's very common. Of course, an easy decision for someone, sitting in a Western world is to say, oh well, these data are not generalizable, so we're not going to use it. But what if that country turned out to be a very big market someday? So those are the things that we all need to take into consideration as well. And then the second heterogeneity is that, of course, in different countries, there are ethnic, different ethnic difference. So in a very broad sense, that's a biological biomarker issue. And then lastly is that, even though there are, the clinical society will put forward, practice guidelines. So for this type of lymphoma, this is how it's supposed to be treated. But as my previous speaker all mentioned, that, real world practice, vary tremendously. So even within the same region, but also then across different countries. So I believe this is my last slide, and that is, oh, sorry, I have another slide. So, now that we're in 2023, so what are the things that we face? This is already mentioned by the first speaker, is the clinical relevance of the historical data. So if you're considering a historical control, how historical that can be, because if it's too old, then it's ancient as useless. Okay, and then, so that's looking back. But if you're developing a new drug, you need to have some horizon scanning perspective. What's going to happen in the future? So is there going to be a competitor, as the first speaker just mentioned? That is there going to be a competitor on the horizon? As you move forward. So how would that factor into your ongoing study? With an external control arm? But honestly, this is not that different from planning clinical trials. So in my prior job, I was head of a clinical trial center, so I have a ton of experience, interacting with global companies on how do you design an appropriate study in Taiwan, where the practice pattern is so different. All right. And the solution, if I may, is actually what all of us are working on right now. So we epidemiologists is the last bullet point. And that is, if we have good data, we develop good data sources, and therefore that external data sources in a contemporaneous fashion can provide a lot of insight into how you maybe modify your ongoing design, and then, to take it from there. All right, this is the last one, and I would go back to a monumental article published by Dr. Janet Woodcock of the FDA several years ago. That was in England Journal 2017, and she proposed the term actually, that term has been used, but she highlighted the term a master protocol. And that is, for a single disease, and I believe some of my previous speakers also mentioned that already, is ideally for example, like a multiple myeloma or a B-cell lymphoma, that is some sort of a very big, wide, global registry going on. So that it could be used to design studies. So it can be used to design clinical trials, enroll patients, but also to address the heterogeneity issues that I just mentioned. Actually, I went back to that paper by Dr. Woodcock, and actually in figure 3 or table 3, actually there are 2 points that are relevant to us epidemiologists. And the one concept is the shared control patients. This also appear in the FDA guideline, that control group from existing control trials, can serve as external control, in this setting. And then lastly, Dr. Woodcock also used this term, the natural history cohort. All right, thank you very much.
Hi, good afternoon. My name is Kathy Laro. I'm an epidemiologist at the U.S. Food and Drug Administration in the Oncology Center of Excellence, working in the real world evidence group. And I'm excited to be here today to discuss a regulatory perspective on the use of externally controlled trials in oncology. I just want to note up front that because this is an international conference, some of the regulations I plan to discuss are specific to the U.S. However, I think the design, considerations are more broadly relevant. Conflicts. I want to highlight up front a recent draft guidance that was published by the FDA's Office of Medical Policy entitled Considerations for the Design and Conduct of Externally Controlled Trials for Drug and Biological Products. This has been mentioned by several of my colleagues today. The guidance serves as a resource to understand FDA's thinking on this topic, and I'll reference it periodically throughout my discussion. So here I have a very high-level summary of general requirements for drug approval in the U.S., starting kind of big picture. So the requirement that new drugs show effectiveness is based on the 1962 amendment to the Federal Food, Drug, and Cosmetic Act, and this law requires substantial evidence of effectiveness and specifies that this evidence must be derived from adequate and well-controlled clinical investigations. The FDA examines this evidence in the context of the disease, the study design, the endpoints selected, and the strength of the evidence to determine whether the data are sufficient to define an appropriate patient population, as well as to provide adequate information to enable safe and effective use. Regulatory decision-making considers both the statistical robustness and the clinical meaningfulness of the treatment effect. So how do externally controlled trials fit into this paradigm? So to demonstrate efficacy of a treatment, a randomized trial is always preferred when feasible. Randomization, as we know, ensures balance of potential patient disease factors that can influence study outcomes. And clinical trials can have other beneficial design features as well, such as blinding, protocol-defined clinical follow-up, independent central review, as was discussed earlier. In oncology, single-arm trial designs and response endpoints, as also was discussed earlier, may be used to establish efficacy of an investigational agent. response rate is a marker of drug activity because malignant tumors typically, as we know, don't regress on their own. And this endpoint can be interpreted in single-arm trials for monotherapy oncology drug regimens. Externally controlled trials may be considered adequate and well-controlled investigations, but in the absence of randomization, there can be challenges in distinguishing the effect of the drug from the usual course of disease or other sources of potential bias. I do want to also note that trial design alone doesn't determine whether the evidence is sufficient, and poor execution can render a trial of any design to be not adequate or not well-controlled. So there are some contexts in which the use of an externally controlled trial may be more appropriate. So for example, when there are feasibility challenges to conducting a randomized trial, ethical concerns, or when there's a clear lack of clinical equipoise. And given this context, areas where these externally controlled designs may potentially be considered for regulatory submissions are in pediatric populations, rare diseases, patient populations with significant unmet medical need, rare molecular subgroups, or patient populations commonly underrepresented in clinical trials. An externally controlled trial may provide valid inferences about treatment effectiveness under the following conditions, and this is from a recent rare disease guidance. The natural history of the disease should be well defined. The external control population identified should be comparable to the treatment group. So ideally, the control should represent the treated patients in the trial had they received the standard of care. Any concomitant treatments with potential to affect the primary study endpoint should not differ substantively between the treatment and the control arm. And finally, results should provide a compelling evidence of a change in the established progression of the disease. And this can include, for example, a large effect size. The suitability of an externally controlled trial to support a specific application is highly dependent on the clinical setting. And as such, you'll hear this from anyone from FDA. We always encourage sponsors to consult the FDA early to try to discuss ECTs and whether this or other trial modernization approaches may be appropriate in a specific clinical context. When it's been determined that an externally controlled trial may be appropriate as a study design choice in a given clinical setting, the next step is to determine whether we can design a study that is actually considered adequate and well-controlled. The FDA draft guidance walks through considerations regarding externally controlled trial design, the importance of selecting a fit-for-purpose data source, aspects related to data analysis, and advice to facilitate FDA review of applications, including external controls. And in the interest of time, I'm just going to touch on a few aspects related to ECT design and considerations regarding the challenges in selecting comparable and fit-for-purpose data sources. The objective of an externally controlled study may impact the choice of the study design. In fact, it should impact the choice of the study design and the source of external control data. External controls are typically conceptualized in two ways, and I think we kind of talked about both of these a little bit in the context of the symposium today. So they can serve as a benchmark, a baseline, or a natural history for a patient population to evaluate cancer outcomes for patients receiving the standard of care. And in this situation, they're providing sort of indirect contextual evidence only. An external control can also use patient-level data to make formal statistical comparisons with patients in a single-arm trial. And this design generally provides a higher quality of clinical evidence of effectiveness because we can control for confounding and we can really compare baseline factors in a more granular way. So careful planning by creating a pre-specified protocol, and this was also mentioned in an earlier session, very important. And SAP can reduce issues in the analysis phase. The protocol should be finalized in discussion with an agreement with the appropriate review division before initiating the analysis to avoid unblinding. There is a spectrum of potential data sources that can be used as an external control. And again, this was previously discussed, so I'm going to kind of rapid fire through these. But they range from existing clinical trial data to real-world data sources and published literature reports. And the study objective really will inform the choice of the data source. So when effectiveness is the primary objective, detailed patient-level information is required in order to ensure that comparisons are valid and that steps have been taken to minimize bias and confounding. So using data from another clinical trial, for example, as an external control can have certain advantages compared to using data collected during routine clinical care, based in part on sort of the rigor of protocol-based data collection. In addition, FDA recommends that sponsors generate audit trails in their data sets and can track access to an analysis performed on relevant data sources. So I wanted to mention that as well. Assessing the comparability of collection or measurement of key factors across the trial and the external control up front can help elucidate potential sources of bias. So it's important to really think about this critically. It can also help us identify where sensitivity analyses and quantitative bias analyses can be implemented to really assess the robustness of the treatment effect, where these differences in the external control versus the trial occur. And so I don't think any of this is foreign to this crowd, but selection bias, confounding, and time-related biases are, three potential threats to study validity that we carefully consider from a review perspective. And I'm just going to walk through a very high-level example of each. So regarding control selection, contemporaneous controls compared to historical controls are generally preferred. Again, as was discussed earlier, especially in clinical settings in oncology where the standard of care or supportive care is changing rapidly over time, and this can lead to increases in survival or improved disease prognosis. And in this setting, use of a historical control would favor the investigational arm. If a contemporaneous control is not available, one option may be to stratify the control arm results by time period and evaluate trends in response or survival over time. If you're seeing, improvements in survival over time, it's an indication that this control arm is not going to work for you. Another challenge that we worry about with observational studies, and by extension, externally controlled trials, is unmeasured confounding. Oncology patients who enroll in trials, we know may differ from those who don't, and these subtle or maybe not subtle differences can confer a survival benefit. Some factors may be measurable, such as ECOG status, although it may be heavily missing, while others are harder to measure or even to define, like socioeconomic status. Using data from another clinical trial as the control can alleviate some of these concerns. However, we would always prefer a robust assessment of potential for bias due to unmeasured confounding be included as a part of any analysis, regardless of what the source of control data is. And finally, immortal time and index date issues can be a challenge, again, as was discussed earlier very nicely, when the index date for the control arm is difficult to define. So for example, like was mentioned, when the trial allows for multiple prior lines of therapy, when there's maybe perhaps an induction and consolidation regimen that precedes the investigational arm that isn't common in the standard of care, or when there's no standard of care and the controls may be receiving, various regimens, physician's choice, for example. And in these settings, it may make sense to conduct sensitivity analyses using additional index date definitions and kind of examining how this impacts the observed effect of the drug, in addition to more sophisticated analyses trying to really understand the impact of immortal time bias. I'm just going to finish up with a few thoughts summarizing the use of externally controlled trials in oncology drug development. So where randomization is not feasible, external controls may be useful to inform drug development. Each externally controlled trial requires careful design and selection of fit for purpose data. And finally, trials that include external controls are complex. I hope we highlighted that in this talk today. And sponsors that are interested in using these designs in their drug development program should contact the appropriate clinical review division early and often. And this is our contact information for our team. I just wanted to flash this up here in case there's any additional oncology RWE questions. And I want to acknowledge my colleagues in the oncology RWE program, specifically Donna Rivera, Paul Klutz, who are here today, as well as others within the agency that are thinking really carefully and thoughtfully about these complicated issues. So yeah, thank you.
Thank you for the presentations. I would like to invite the panel to the front so we can open the floor for questions from the audience. Yeah. Right. Sorry, I hope it's not too uncomfortable on the floor in the back. But the smaller conference does make us better attended. So if anyone has any question, please step up to the microphone and say your name and your affiliation. No, I'm just here. Yeah.
Yeah. My name is Huali from Daiichi. Thank you. Yeah, thank you very much for on this excellent presentation. So I have some question. The first question for the first presenter: how do you handle if you pull data from multiple databases? Patient may be overlap. How did you handle?
Yeah, maybe I can take a shot on this one. So I think, I don't know if you're familiar with the de-identification of the patients. No, the deduplication, sorry, not the de-identification. Everyone should be de-identified. So we have been looking into a technique, just call it tokenization. So that allows you to deduplicate the participants that may potentially exist in multiple data sources. Maybe, I don't know, Paul, if you want to talk about that from your perspective as well. I know you know better.
Yeah, so can everyone, is this working? Okay. Yeah, so there are in oncology a number of commercial EMR providers and I guess adjacent providers who do provide tokenization, which allows you to link different EMRs together. It's I don't know if anyone's familiar with the concept of fuzzy matching, where you can kind of link patients together probabilistically based off of certain demographic characteristics. In some cases, you can utilize social security numbers to do that. And so if you have you know, there are commercial providers that allow you to link those data sets together, and that gives you the opportunity to remove lines of therapy for given patients that are appearing in both data sets, or even prioritize perhaps one data set that has higher quality data on that line of therapy or that patient. So that's generally the preferred strategy for doing this. In some instances, if you're dealing with academic registries or registry-like EMRs, where the data is coming from academic sites, there are instances where the data privacy rules are such that they won't allow tokenization because it requires the data go to a commercial provider. So in that case, it's a little bit more challenging, and you have to develop more of a de novo in-house style of fuzzy matching algorithm. But generally, those are the two strategies that I've encountered in my experience in handling that issue.
Yeah, thank you. I have a second question for second presenter. So you
Sorry.
make some. Can I?
Sorry. Yeah. Sorry. I just wanted to add one more point. If you are dealing with the same type of data, like you wanted to augment the different EMR together, which is probably fine using tokenization, right? To solve this generalizability issue. But if you have a claims data and EMR data or registry data, never augment them together for a single analysis. It's because the data collection curation mechanism is entirely different. So the inherent limitation of the data is different. You shouldn't combine them together. I've seen practices combining open source data, closed data together for some sort of analysis. That will cause a lot of issues. So, you know, in my hypothetical example, I would suggest you to do separate ECA analysis, reporting, you know, the results transparently. So create a package of evidence to show to the regulatory agency about the relative effectiveness.
Yeah. Okay. Thank you. I have second question for second presenter on validation of the assessment of the response. Of course, if we can use this CT scan imaging or the real-world crisis, whatever outcome. I have one question. If what do you interpret if the ORR is lower in real-world cohort, external control arm, but better survival than in clinical trial? How do you interpret? If in clinical trial, best ORR, but worse OS?
Yeah, it's a very good question, and I don't have a very good answer for it. I think you would have to think about the underlying mechanisms which may have contributed to that. It may not be an issue of scan or independent central review, but it may be other effects that contribute to differences in relative ORR rates and relative OS rates. Doing the analysis that I described, condition on available response in one, ORR doing it 2 ways, OS doing it 2 ways, that can give you some insight. But then also think about, do you expect a OS benefit of your treatment at all? Do you expect an ORR benefit? There are other data streams that can help inform that.
Okay. Yeah, I have a last question on the pi zero. Actually, I faced myself with to select pi zero on index day, and on this cohorts in rework data and up only third line. But in clinical trial, they can from third, 4, 5, or 6. So I think Paul presented like 4 methods, but actually at that time, like 3 or 4 years ago, we faced similar issue. But one sampling method, do you recommend this which kind of sampling method? Simple, random, or the other methods, sampling method?
Yeah, so you're asking about selection of random selection, so which are the preferred methods out of those? So I hope I'm answering your question correctly, but there are different variants. I think the challenge with random selection is you generally depend on observing available lines of therapy, which can induce a little bit of selection bias. I guess there are alternate approaches to doing that where you can just select different lines of therapy with using an external probability distribution. So you select one line with a probability distribution of 0.5 and the second line with 0.5. And if a patient doesn't have the line that you wanted to select for them, then they're discarded. So that kind of avoids this issue of bias. So within that framework, that's one option you could consider. But if it's a matter of trying to benchmark your data against the external control arm, or sorry, against the single arm trial, then oftentimes waiting on a limited number of factors when you're sampling the random lines can help you achieve that, because then you can sort of develop a natural history cohort that is reflective of some of the baseline characteristics of the clinical trial, so that can be advantageous. I hope that answers your question specifically on the point of choosing different variants of lines of therapy at random.
Yeah, thank you. Actually, we did stratify sampling and we published in Journal of Lymphoma. Yeah, okay. Thank you.
Anne-Marie Meyer, NCIN, a question for Christian. I was so glad to see that analysis. I've been waiting for that analysis on the simulation around the bias. And, with against the conditioning on the scans, because I can tell you that the concern I have in industry, and I've debated this with some colleagues at a great length, you have a risk of immortal time bias, right? And you have to pick the bias, and I'd rather be accused. I mean, you showed that the bias is going by not doing it, the bias is going in our favor, right? So I'd rather pick another bias that is being more conservative, right? And to that point, I think there's still a risk of immortal time bias and I can tell you that the lags and the availability of scans plays a big role, and that is not systematic in the populations. And if we're thinking about, and again, we're thinking about the external control and modeling that as a design, shouldn't we also be thinking about the scan intervals from the trial protocol? Because again, that person time at risk and really making sure that we're not, stacking it in our favor with regard to immortal time bias. Shouldn't we also be thinking about that person time and the design from the trial that we're emulating with regard to those intervals, not only having it, but also the interval?
You mean the intervals between the scans?
Yeah, the protocol intervals defined by the trial.
Yeah, very much. In reality, most of the time you will only have one follow-up scan time point, so you don't even have those multiple. So it's pretty much just one snapshot, and you hope you're capturing the patient's best overall response. That's really a limitation, and not sure how to overcome that really. Yeah. Regarding conditioning on scans, you're right that there are additional biases that I may have not addressed in full detail. Selection bias is a concern because patients who have scans may differ from patients who don't have scans. And that may differ by geographic region. It's possible that treatment guidelines or monitoring guidelines in the real world differ depending on where you are. Some may recommend scans, but scans are costly. So in some areas, scanning patients in terms of monitoring treatment response may not be routinely done. And then you have to think about in areas where there's discretion, a physician can decide whether to scan a patient or not. The patients who do have scans will likely differ from patients who don't have scans. If you are in an area where it's routinely done, you probably have less of a bias there. So really understanding the mechanisms that underlie who gets a scan and who doesn't, and then analyzing patients, comparing baseline characteristics of patients with scans without, really helps understand the data.
I totally agree. And that's where the unmeasured confounding and people are like, oh, but payer, should we be conditioning on insurance status? Because let's face it, the medicated or the uninsured or underinsured patient in that real world data set is not exchangeable with a trial patient. And also, again, the EMR pipelines with regard to the availability of the scans, if they have a fully integrated RIS and DICOM, that's a really different scenario.
Next, is this working? As now? Okay, this is working. Yeah.
Hi, Mugdha Gokhale. I work for Pfizer. So, Arnold had mentioned in his presentation about, often in hematologic malignancies, we have combination treatments, right? So drug A plus standard of care was the example you used. If we have a situation where we have drug A, or trying to combine it with a novel therapy that's going to come. So drug B, which is also not approved. So basically, we're looking at drug A plus B combo, which is both are under development. And in such a case, I wonder, in the interest of accelerating clinical development timelines to reach the patients faster, would you still consider a single arm trial? And what do we compare it to? Because drug B is maybe like approved for later lines of therapy, but we don't have real world data for those in the line that you are trying to develop it in combination for. In reading the literature, there are options like digital twins and whatnot, and I'm still myself trying to absorb things around that. But I'm just curious, any thoughts, any considerations? How do we even begin to think about that?
Well, first of all, I'm glad I made the disclaimer up front that I don't have a solution for everyone. So I guess I opened up a can of worms. And the scenario that you described, it's what I thought about. But I thought about it in a hypothetical manner. But what you describe seems to be a real case that so I guess what you're mentioning is that, okay, you have standard therapy. And instead of having one novel drug, you have a combination of two novel drugs. And I guess that's the challenge for regulators. So I would then refer to kind of no, I'm not putting you on the spot. I'm just saying that, okay, maybe this is the time to have that early and frequent conversation with the regulator on how to pick, because that's honestly, that's my solution. Once again, I apologize. I don't mean to put you on the spot, but
yeah, I will just say that that is a challenging situation for sure. And I think part of the problem and why you're asking this question is because it's tough to sort of understand the contribution of each of the drugs and whether both of the drugs are working together, or if it's really just one of the drugs and the other one's not doing anything. Yeah, that's a tough question. I also don't have a great answer for you, but definitely challenging in a single arm setting.
Thanks for that. At least, that adds to my, I guess, mental fog where I'm like, I'm not the only one in the fog. Everybody is dealing with the same thing. So thank you for that. And very interesting presentations by all. Thank you.
Let me add to the question for maybe Arnold and Catherine. So what's your understanding of the standard of care? For example, is off-label use standard of care? Right. Sometimes there are guidances on the treatment guidelines, and then sometimes off-label use can be a big proportion of the actual treatment landscape. So if you pull out real-world data of ECA, and then the treatment distribution can maybe not what you expected from like guidance perspective. So would you drop the off-label use or you include them?
All right, so I'm going to stick my head out, and then being from a public, from a practical kind of perspective, I wouldn't consider on-label, off-label. I will be driven by the empirical data. So if there seems to be an effective regimen that is off-label but is widely used, then I would consider it to be a legitimate comparison group, from a clinical perspective, because that help answer a clinical practice question. Now, if you are seeking like a regulatory decision, then once again have to refer back to the regulators, to comment on what would be the appropriate. At the end of the day, I don't make that decision. It's the regulator plus the advisory committee, discussing that topic.
And no pressure, Catherine, just.
No, I guess my I would have like follow-up questions for that. It's really, really dependent. But my question would be, why are you trying to use something off-label as a standard of care? Is it standard of care because those patients are too sick to receive an approved drug? Like, is there a reason? So I guess there's probably a lot of like intricacies to answering that question. And I would always defer to the clinical review team as to what the appropriate standard of care is in a given setting. I think as the epidemiologist, that's not really my purview. So that would be my
right. So I think my question was primarily for some of the rare conditions, especially the ones without any standard of care that's been approved. So once you pull out the data, so the treatment is just everywhere. So would you consider in that situation that's reasonable standard of care for that condition?
Yeah, if there are no approved therapies, I think it's a physician's choice situation, presumably.
Okay. Thank you. Next.
Yes, Michael Kelsch, formerly with Amgen and now with Epid Strategies. I agree, very nice presentations that were all very interesting. And I know, Arnold, I promised you a question, but I'm not going to ask you a question. So you're off the hook. But I do want to ask a question to Paul about time zero, because I think, or just maybe more of a comment. One thing, if you're looking at an indication that's like second line or third line, why don't you just go straight to that in your definition of selecting which line? You didn't quite mention. And then the second thing is, we were dealing with this issue a number of times, and you can do sensitivity analysis on your decision of whether you picked the last line or the first line, or you picked the random sampling approach, which I kind of favor over taking all of them that you recommended, simply because I worry about, dependency when you're taking two measurements from some and not from others. And is there a selection in that reason? They're getting more intensive care or something. So just thoughts, or I'm curious to know what your response is to that.
Yeah, I think maybe the lost context is that I was really considering a setting where patients could be on a third line or subsequent line of therapy in the clinical trial, in which case, you might want a similar population. So in that case, it could be useful to look at multiple lines of therapy where the patient is not just on the third line of therapy, but could be on fourth, fifth, and so on. But if you're right, if you're really targeting a patient population in a trial where these patients have failed 2 conventional prior lines of therapy, and now you're really narrowing in on that third line of therapy, I would agree. It makes sense to find the first line of therapy that corresponds with that, because then you have similarity in eligibility criteria. So, and then I guess with respect to random line versus all lines, I kind of briefly noted that when you select a random line of therapy, you're kind of doing the sampling process once, but you could also do this via simulation many times, in which case, you know, in one iteration, you might select the third line, and then in the next, you might select the fourth. And if you do that many, many times via simulation, you arrive at probably a similar conclusion as you would had you selected all lines of therapy. So I think that there's a bit of an overlap between those approaches if you generalize a little bit.
Yeah, and I think if you did a sensitivity, you would know, does this matter?
Yeah.
Or you would understand your data a little better that, you know, defining a third line gave a biased result and or whatever. I mean, but I think it could help and just explore all the data you have and the impacts. So thank you.
We are on time, but we can certainly take one more question. Everybody's still here, so thank you for staying with us.
Yes, thank you. Jun Yuan from Janssen. I really enjoyed the presentation here. It's a really sort of a timely topic, a lot of discussion, even within the industry, talk about the external control in the situation when randomized trial cannot be designed. So my question to Paul, again, similar to the previous question in terms of time zero. We're talking about emulating the clinical trial, right? So in the clinical trial, you don't ask patients start second line therapy. That's one to zero. The 0 day for the single arm study is the interventional drug. The other drug is not the zero we define. Although it is a straightforward for the line of therapy, you can define as the index date when it's starting, or even for the last line therapy starting, it is a straightforward. But in terms of estimate, this is not estimate the single trial try to estimate, because some patients in that population, they could be already in the middle of the therapy, you just add on the existing therapy. So from that perspective, if you do have a contemporary external control group, potentially can be used as an external control arm, why can't be used calendar date? So that calendar date was similar to the trial when the patient being enrolled while it's in clinical trial setting, they're not enrolled, but they're patient and you're controlling all the existing standard care therapy, whether it's in the middle or it's just starting, that could that be a potential as control arm, while the starting zero is not as straightforward as the proposal you have proposed, but from estimate or emulating trial situation, that seems one potential option. So happy to hear your thoughts.
I'd love to talk more with you about this, but I think if the question is if you have access to a contemporaneous external control arm, does the story change a little bit? And I think you're right, it probably could. So if you could then look at lines of therapy over a contemporaneous screening or enrollment period, does it hurt to pick the first or a random line of therapy? I haven't really explored that question, but you know, I'd be happy to talk with you about that. So I think I'll probably pause there just with time, but it's a very valuable insight. Thank you.
Thank you. Maybe we can thank our panel for the last time for the presentation. And thank you everyone for being with us today. Hope you enjoy the rest of the conference.